Thank you, Will, and good morning, everyone. It is truly a pleasure to welcome you to MARCO's 2023 Mid-Atlantic Marine Debris Summit. I'm honored to stand in this room filled with dedicated individuals who share a passion not only for preventing and reducing marine debris, but also for safeguarding our marine ecosystems. First and foremost, as Will teed up so eloquently, I do want to extend a heartfelt thank you to the incredible planning team whose commitment and tireless efforts have brought us together today. If you're on the planning team, can you just raise your hand? A round of applause for these committed folks. As many of you know, MARCO is the regional ocean partnership for the Mid-Atlantic. It spans the states of Virginia, Maryland, Delaware, New Jersey, and New York. And it's at this regional level that we discover the sweet spot where nuanced local challenges meet larger scale solutions. Operating at this regional level allows us to better understand how to tailor big federal and national and sometimes even state priorities to meet the unique ecosystems, cultures, challenges, and opportunities of the ocean users and coastal communities here in the Mid-Atlantic. Today, as we delve into the presentations and discussions and insights that will unfold, let us keep in mind that the importance of regional collaboration is here. It's where the magic happens, where federal objectives meet these nuanced realities of local communities, and where hyper-local programs are elevated through collective regional efforts. Today, we'll hear and see presentations from scientists, decision makers, community leaders. We'll learn about the great work they're doing to address the pervasive issue of human-made debris in the ocean and coasts. But also importantly, today is about bringing all of these individuals with diverse expertise, jurisdictions, and organizations under one roof, united by a shared purpose. It's also about highlighting the incredible work happening at this local level, like what you'll hear about from our keynote speaker this morning, Uganda Sample Jones. These local efforts are the heartbeat of our shared mission, and they deserve not just acknowledgement, but celebration. So, as we embark on this journey of insights and collaboration, let us remember that our strength lies in unity and our success hinges on our ability to connect, learn, and act together. Thank you for being here, for your commitment to this crucial cause, and for the strides that we will undoubtedly make together in the pursuit of cleaner, healthier oceans. I am now thrilled to introduce Kristen Fidler, the Assistant Secretary for Aquatic Resources at the Maryland Department of Natural Resources. She joins us today via video. Kristen is a Maryland native, having graduated from University of Maryland College Park, as well as Maryland Law School, with a concentration in environmental law and policy. Prior to joining DNR in April, Kristen served as the Director of Harbor Development at the Maryland Port Administration, overseeing the maintenance of the federal navigation channels serving the Port of Baltimore. In 2020, Kristen was recognized by the Alliance for the Chesapeake Bay as a Maryland watershed champion for her contributions through innovative thinking and the development of inclusive and impactful partnerships to advance stewardship in the Chesapeake watershed. And I just do want to note that Maryland, the state of Maryland, is the MARCO board chair for the next 2 years, and so we are thrilled to be hosted here in Ocean City, Maryland, thrilled to have Kristen join us via video. And again, thank you all for joining us today.
Good morning. Good morning, everyone. Thank you so much, Ms. Bristow, for the opportunity to be a part of this summit and this convening of so many partners and experts. Again, I'm Kristen Fidler. I serve as the Assistant Secretary for Aquatic Resources at Maryland's Department of Natural Resources. And on behalf of the Governor, Governor Moore, and Secretary Kurtz, I want to extend a warm welcome to all of you who are visiting from out of town. Welcome to Maryland. Personally, this is my favorite time of year to get down to Ocean City or one of the Delaware beaches nearby for a visit, so please know how disappointed I am to not be in person with you today. But I did want to offer a few remarks to keep in mind as you spend the next 2 days listening to experts who spend most of their time taking a really deep dive into all of the various issues surrounding the marine debris challenge. Try to keep this one concept in mind, that while the issue challenging us may seem overwhelmingly enormous and daunting, and even that much more complex because it's comprised of issues that range from as small as tiny nanoplastics to as large and cumbersome as abandoned vessels, so too will the solutions need to be diverse and varied. No action will be too small to make a difference, and certainly while there's more than enough work to go around for each one of us, so too will the collective effect of our partnerships and amplifying each other's efforts will also be what helps to turn the tide on this very challenging matter. And so I guess what I'm trying to say is that tackling this issue in all of its nuanced facets will take all of us, whether we're representing a federal agency, a state natural resources agency, nonprofits, local organizations. So while it is that large and it will take all of us, I don't want it to be so overwhelming and daunting from the outset that we don't also focus on and recognize the role and the importance of small scale, local, and even hyper-local level changes and impacts and actions. In Maryland, we are absolutely committed to working together with our regional, our tribal, our federal, our nonprofit, our sister state agency partners to innovate, to leverage funding, leverage resources, and really to break down silos so that our individual efforts can more effectively and efficiently go further. We must ensure that as we continue to move forward with research, with implementation of the findings of that research, with educating students ages K to gray, as we like to say, and looking for new opportunities to collaborate, that we continue to include all voices and all communities in that discussion. And that really is what's so exciting about this summit over the next 2 days of listening and learning and networking, sharing, building new partnerships. You'll be hearing from researchers, from organizations, and experts at the forefront of this field. And they'll be identifying new ways to look at marine debris, new ways to approach it, how and where it starts, its impacts on various species of wildlife, ecosystems, economies, public health, the various public perceptions around marine debris, challenges, barriers, opportunities to engaging with the public, how to conduct effective outreach and education. I'm excited about this summit, and for all of you that are there right now, I really am confident that dots will be connected over the next 2 days, whether they're tiny nano dots or large-scale aha moments where synergies are ignited. As you all well know, whether we're talking about wildlife on land, in parks, or in our oceans, or in the Chesapeake Bay, it is all connected. Whether we're talking about climate change, sea level rise, warming temperatures, or shoreline restoration, erosion, water quality, diverse habitat in water, it is all connected. Whether we're talking about the health of our urban neighborhoods, investing in flood prevention and infrastructure, sediment in our streams and local rivers, the multi-agency, multi-jurisdiction restoration of the Chesapeake Bay and our commitment to that, or whether we're all just talking about and marveling at how majestic this Atlantic coast shoreline really is. It is all connected. This ecologic, economic, environmental, and public health risks posed by an ever-growing marine debris problem are too great and will only be exacerbated by stronger and more intense storms, infrastructure failures, and a lack of awareness of the problem itself and how it and we are all connected. This problem spans sectors, industries, communities, agencies, species, and so it requires solutions just as diverse and varied. Solutions that will be stronger and more effective when knitted together by cross-sector partnerships, by coalitions big and small, by industry, technology, all of this only to be limited by the innovation, the creativity, and the imagination of each of us working on it, alone and together. So in closing, thank you all so much for the work you're doing now and will continue to do moving forward. Thank you for dedicating your time, energy, and passion to such an important matter, and for taking this time today and tomorrow to learn, to share, and to think outside the box. We will get it done because of the work you are doing, and we're grateful for it. Thank you so much, and enjoy Ocean City for the next 2 days.
And we extend our gratitude to the Assistant Secretary Fiddler. That was great, and I wholeheartedly agree. Lots of dots will be connected, and I'm very excited to connect this next dot. Now it is my pleasure to introduce Uganda Sample Jones. Uganda is a community organizer in Newport News, Virginia, and the founder and president of Empower All, whose mission is to encourage, expose, and engage people living in marginalized communities to cultivate socioeconomic change. She's one of those people who seems to have a hand in all facets of her community, improving the quality of life and representing the underrepresented voices. And while I don't have nearly the time to go through her very impressive bio, I recommend you all check it out by scanning the QR code on the back of your name tag or on your agenda. She has lots of great experiences, diverse and running all over the page, but lots of very impressive things to be looked at. Anyway. With that, many of us are aware, though, that these issues related to trash have a disproportionate impact on marginalized communities. While it's important to work with these communities, it's also important to understand their priorities and where our goals may overlap so that we can work effectively together. So with a wealth of experience in community engagement and organizing, I'm so very excited to welcome to the stage as our keynote speaker, Uganda Sample Jones.
Good morning, ladies and gentlemen. Esteemed guests and passionate advocates for a cleaner, more sustainable future. I am truly honored to stand here before you today as we embark on this pivotal moment. My name is Uganda Sample Jones, as you see there, and it is a unique opportunity for us to come together to share our insights and to forge collaborations that will shape the course of environmental stewardship in our region. I must confess, this journey to this podium here has been a mix of delight and nervous feelings, a feeling many of us may share when you're faced with the responsibility to inspire change. But when William first extended the invitation, I found myself questioning my place among the distinguished individuals here. I was like, okay, could there be a mistake? Or why me? But as my daughters and I and my mother, we rode through Ocean City last night and yesterday, I witnessed the waterfront that binds us together. And I am reminded of the seeds that were planted in me as a child, where I had a longing for a world more beautiful and cleaner than the one that we inherited. My family roots run deep throughout the Eastern Shore of Virginia and Maryland. I remember summers and holidays spent with my grandparents that encouraged a deep appreciation for nature and unpolluted green spaces. I was raised in Newport News, Virginia, first generation. My parents, they instilled a strong sense of responsibility within me and my sister, and also a value for leaving spaces cleaner than where we found them. My desire to keep our environment free from trash was cultivated, and I developed a strong discipline against littering. So please join me as I extend my gratitude to my mother and my two daughters for joining us today. My mother in particular, she played a crucial role in shaping my environmental consciousness. As I prepared my notes today, I reminisced about a time when she used to take me and my sister to the waterfront, the beach in Newport News. And when it was time for us to go, she would ever so cleverly employ us to clean up our space and make it cleaner than when we left it, than when we came, with the promise of giving us ice cream after the trip. My parents kept my sister and I engaged in activities like Keep America Clean, Keep America Beautiful, school recycling projects, and these fostered a commitment to not litter and keep our surroundings clean within myself. As I entered adulthood and I started my own family, the tradition of mindful living continued. Community cleanups became a family affair, reinforcing the importance of keeping our spaces free from trash. Isn't that right, girls? When I returned to Newport News in 2008, I was confronted with the stark reality that my community that raised me had changed drastically. The buildings that were once magnificent in structure had been abandoned. There were spaces where Businesses, we were able to have conversations and fellowships that led to partnerships and reliable programs, and they no longer existed. And what was most heartbreaking was the once beloved waterfront had fallen victim to neglect. Now, this fueled my determination to make a difference. I joined the PTA, and I was able to organize school beautification efforts and cleanups with the students. It was there that I learned the basic rules of engagement. Like, students and parents will show up on Saturday mornings as long as you make it fun with a DJ and definitely donuts. So nevertheless, I felt that wasn't enough because I would see people throw trash out of their cars and it would absolutely infuriate me. The realization propelled me beyond the physical debris cleanups, and it was very evident that we had to address the mindset that allowed people to litter, and addressing the systematic issues that were there as well. My community was marginalized over the years by resources and education leaving the east end of Newport News and reestablishing in less racially integrated neighborhoods of the northern Warwick County. This left a dense population of poverty, poor maintenance for public housing sites, and high crime. It was clear to me that the community cleanups are not just about physical debris, but about cultivating ecosystems of positive change throughout the entire city. So my involvement in initiatives like the Neighborhood Watch Coalition and the Choice Neighborhood Initiative revealed the power of uniting individuals from diverse backgrounds with a common goal and an aspiration to make things make a change greater than their individual selves. So as we gather here today, we must acknowledge the critical role of intergenerational wisdom, stories, and teachings. Connecting those with the knowledge and resources, like y'all here, with the marginalized communities is pivotal. Communities that, due to historic inequities and injustice, like redlining and toxic brownfields, find themselves at a disadvantage, deprived of educational resources and connections to address the environmental issues that plague their doorsteps. I live in Newport News, and we have a coal terminal there. And the residents, although we have smut and dirt on our houses, they really don't pay it any attention because the education and the resources aren't there to be able to teach us how to do something about it. So environmental justice demands our attention, and it is a collective duty to address historical disparities. Our waterways are the lifeblood of this planet, and they defy individual control. So we can't just put a water meter on the Chesapeake Bay or the vastness of the Atlantic Ocean. The responsibility of safeguarding this precious resource lies with each of us. As we collectively navigate the challenge of reversing the detrimental effects of water and air pollution, today our summit's objectives reflect the commitment to inclusivity, justice, and collaboration. Our first objective calls for the fostering of meaningful collaboration among stakeholders, encouraging interdisciplinary collaboration, and strengthening partnerships. The power of collective action is supreme, and by merging our resources and expertise with the people who are most affected by the problem, we magnify our impact. As we explore solutions, let us ensure that our communities or no community or individual is left behind. So often, experts and organizations will come into our communities and tell us what needs to be done. That's not the way. Include the parent and the child that walks through trash cans, that walks through clogged drainage ditches to get to school, or the teenager that has ideas on how to erase the effects of broken glass theory. By acknowledging and dismantling these discrepancies and barriers, we pave the way for innovation and effective solutions. Knowledge sharing is a powerful catalyst for change, and by showcasing success stories, we inspire others to take the mantle for environmental stewardship. As we delve into the complexities of shared challenges, let us not forget the historical injustices that have shaped the landscape of environmental disparities. I was fortunate enough to work on a research project with Professor Johnny Finn, associate professor at Christopher Newport University. This project creatively brought together the academia community with the people that were affected by the pollution and environmental disparities in Newport News by actively engaging us in every part of the research phase. Professor Finn and his students conducted interviews with over 100 participants throughout the Hampton Roads metropolitan area. Dozens of participants were armed with cameras, little disposable cameras, and they captured hundreds of images and places that were meaningful to them within the community. By doing this, the university was able to gain the trust of the community, which revived the pride and foster a greater sense of community. I highly suggest you check out Professor Finn's research project, Living Together, Living Apart, Geographic of Segregation in the 21st Century, to explore in extensive detail, of the ongoing socio-environmental impacts on how the legacy of redlining and segregation impacts our communities today. Redlining and brown toxic fields have left lasting scars on marginalized communities. To truly advance environmental justice, we must confront the disparities head-on and work towards a future that is where every community has the resources and support needed to have a cleaner, healthier environment. In the spirit of collaboration and shared responsibilities, let us draw on the inspiration from the wisdom in the generations past, as well as the fresh perspective of our youth. It is in these spaces that we command every day, that each representing your own unique nexus of knowledge and influence, that we have the power to spark transformative ideas. I am confident that at this Mid-Atlantic Debris Summit it will be a vessel for innovation, for forging a path towards a more sustainable future. So let us please engage in thoughtful conversations, foster collaborations that transcend boundaries. Talk to someone that you don't know. Talk to someone that doesn't look like you, that doesn't have the same experience, and emerge from this summit with actionable strategies that will leave a lasting imprint on our environment and communities that we serve. I want to thank you so much for this journey towards a cleaner, more sustainable future, and thank you so much for all you do.
I'm very pleased to introduce our two speakers for the environmental implications section. We have Christine Knaus of the University of Maryland and Todd Tillman of the University of Delaware. Christine?
Good morning, everyone. It's great to be here. I see some familiar faces, so it's great to see you all. So today I'm going to be talking, so I'm from the University of Maryland Center for Environmental Science, so just down the road in Cambridge, Maryland. If you ever have a chance to come visit our lab, we give some great tours. We have a lot to see. But today I'll be talking about impacts of microplastic ingestion on oyster larvae and if there are implications for our oyster restoration efforts in the Chesapeake Bay. So first, I want to say a huge thank you. None of this work would get done without the oyster hatchery crew, or Katie McFarland, and a lot of these other people. It's a lot of work to try and get oysters, healthy oysters, to spawn and live in the lab. So thank you to all these people. So I'll start here. In 1955, this is Life magazine's story called Throwaway Living. So they're celebrating the proliferation of plastic and how disposable plastics are going to help everybody. We don't have to clean anything anymore. We can just throw it all away and it'll all be fine. Women don't have to be in the kitchen to clean anything anymore. Life is going to be great. And so we know that, and it was touted as a huge revolutionary step, and it is, and it's provided a lot of positive impacts, but as we know, it's also provided by creating so much, it is can be detrimental. And you can see on the right, this is a graph of the historical and the projected annual plastic production. And it's increasing exponentially if we stay as a business as usual. So when we think about plastic pollution, we normally think about images like this, larger plastics, lots of a tire reef, and it's everywhere from the bottom of the ocean to the top of the highest mountains. And for this session, like was mentioned, we're going to be actually focusing on microplastics. So hopefully I'll be setting the stage for the rest of the speakers of this session is what are microplastics? What are we talking about here? So, is this? Yeah. It's hard to see. Okay. So we are talking about anything from about 5 millimeters, which is about the size of a pencil eraser, down to smaller than the diameter of human hair. And so it's a big size range, but very small, and it can be smaller than the naked eye. And so other sessions today, you'll be hearing about some of these other kind of sizes. But when we're talking about microplastics, this is sort of the size that we're talking about. And there are lots of applications of plastic, clothes, furniture. You can still have microbeads in certain products, even though there was a ban on some of them, they're still allowed in other products. Plastic is in tires. Plastic is used in agriculture. From washing your clothes, it gets into the biosolids, and those are applied on farm fields. There can be leakage from landfill. There are a lot of, because of all the uses and applications of plastic, this creates a lot of sources and a huge variety in the type of plastic that you see. And so this is a diagram kind of showing a lot of the different inputs from all those different kinds of sources and the diversity that we see. And so because of this, we get huge differences in abundance, a huge variety of sizes, like I was just talking about, lots of different shapes, from fibers to beads to fragments. We can also, there's lots of colors. In the environment, there can be, so that ages, so the sunlight and wave action and microbes can change the environment, the microenvironment on those particles. And so all of these things can affect, and also the chemicals that are in plastic and the chemicals that are in the environment that stick onto the surface of plastic. Those all make this a huge contaminant suite. So it's not just one thing, it's many things. And all of these characteristics can affect how animals interact with these particles. And so if they're going to be available, some of them to eat, if they're in the size range of, let's say, oyster larvae, or not, if they're the color, if they're the shape that they like. So all of these things are important to keep in mind when we're thinking about exposure, impacts, and risks. So what do we know about zooplankton ingesting microplastics? So the image on the left, all of the glowing green particles, those are introduced microbeads in a lab setting to lots of different types of zooplankton, and you can see that all of them eat plastic. And on the right, there's also, it's hard to see some of the colors, but there are also lots of microbeads in oyster larvae. So that's so we know that, and by now, organisms ingest a lot of microplastics. We also know that for oyster larvae, high concentrations of microplastics can affect different things, like their development, their swimming behavior, their swimming speed, and also their fertilization and other steps of development. Now, in the Chesapeake Bay, oysters are really important ecologically, economically, and culturally. The population of the bay is less than 1%, and because of that, there's been a huge effort in time, money, and resources to help restore the population. Also, larvae are the most sensitive life stage. They are free swimming in the water column for about 2 to 3 weeks. They're actually in the water and not on the bottom. And then also, in terms of microplastics, there's very, very little research on oyster larvae. So besides my work, there's about 4 or 5 papers globally looking at exposures and impacts on oyster larvae. And that's, I think, just because, like I said in the beginning, it's very hard to culture these in the lab and keep them healthy and alive for longer periods of time if you don't have the support from some other researchers. And because of the huge effort to restore the population in the Chesapeake Bay, when I started my PhD, I was thinking, are microplastics and microplastic pollution something that we have to think about when we decide where we restore oyster reefs? Can we do it in urban centers? Is it going to matter? Do we need to do it in other places? Because the idea of a restored oyster reef is that it's self-sustaining oyster bars. So you have lots of larvae that will seed, itself in other bars. And so is this something including diseases, salinity, access, kind of all of these other factors that we need to consider in restoration? So this is the life stage that I focused on. And so in thinking about that question, is this really important, something we need to think about? I came up with some research questions. So oyster larvae don't just eat all the time. There's a certain concentration so they eat algae when they start, where it's efficient for them to eat. So what's the water concentration for Crustacea virginica is the eastern oyster, the one that we have here. What's that concentration for microplastics? Is it the same as algae, or is it not? And are oyster larvae selective? So if there's lots of food and a few microplastics, do they ignore the microplastics and eat a lot of just the food? Or does it matter? So what of that interaction? How are they selective in the particle types that they ingest? When do they reach a saturation? How full? How many beads can they eat at a time? And then if all these happen, if they interact with and ingest and maybe ingest a lot, does it affect them? And then the last thing I looked at was, is there an effect by the different plastic types, or is it all the same? And so I started with the first ones, is seeing if they eat microplastics and how many. So I took oysters and polystyrene microbeads, a huge variety of concentrations, compared with a mix of huge varieties of concentrations of food. So are they selective or not? And expose them for 24 hours. So just allow them to feed for 24 hours. And it turns out that oyster larvae eat a lot of microbeads. And so this is a very classical saturation curve. So on the x-axis is microbead concentration, and on the y-axis is the number of beads per individual. And you can see that, so it starts at just after 0, so it's 20 particles per milliliter is what we're working in, and then it goes all the way up to 20,000 particles per milliliter. So a huge range. We would never see, or we think and hope we would never see 20,000 particles per milliliter in the environment, but just to kind of see how they interact and how much they eat. So they reach a saturation between 10,000 and 20,000 particles. So meaning that as microplastics in the environment of this size, oyster larvae will keep eating until we get to those kinds of concentrations. So that's really important to know, that as we get more plastic pollution and more microplastics, they're going to continue to eat more in the near future. The other thing that was interesting about this was that microplastic ingestion didn't depend on food availability at all. So they weren't selective against microplastics and ate algae. The only thing that mattered for how much they ate was how much was there. So those are 2 important things to learn from this. So the next thing was looking at impacts. So now we know that they eat microplastics and generally how much they eat. Does it affect them? So we looked at the same thing, polystyrene microbeads, for 6 days. and looked at different endpoints like growth, respiration, carbon assimilation, so algal ingestion, and then of that algae, how much they actually built into their bodies, so that's carbon assimilation and respiration. And good for us and oysters, with the polystyrene microbeads, there was no change from the controls in terms of anything that I measured. So algal ingestion, the carbon assimilation, respiration, and growth, even at higher concentrations. So that's good news. However, microbeads aren't the things that we see the most in the environment. So I wanted to try and make something, a particle type that was more realistic and more environmentally relevant. So with some colleagues at the Cooperative Oxford Lab in Oxford, Maryland, we developed a way to make very small and standardized microfibers, so I'd love to chat about that because that was a fun project, to be able to get particles that were small enough for oyster larvae to ingest. And these are not commercially available products like microbeads are, so this was a fun challenge. So we created some very small microfibers in polyester and nylon, and did the same experiment exposing oyster larvae for 6 days and looking at the same endpoints, so growth, algal ingestion, carbon assimilation, and respiration. And I don't know if this will play. So this is a video of an oyster larva. Let's see if it plays. The sort of orange glowing piece is a fluorescent microfiber, and you'll see it start to move. So we can tell that it's in the gut because oysters digest, they have a style that spins. So you can see the fiber that's spinning inside the gut. So, and this oyster is alive. And the red particles, those are the algal cells. So you can see, and then it zips off. So that's just a cool video to know that they are actually not just bringing them in and then spitting them out, which oysters can do. They're actually, the particles are actually going through the digestive tract. So still, oyster larvae were able to handle these particles pretty well. There wasn't an effect on algal ingestion or carbon assimilation. There were slight decreases in respiration rates. There was only a decrease, well, really it was no growth over time, and that's unusual. They should grow over time with really high concentrations of fibers. So oyster larvae seem to handle these particles pretty well. So that's great news for everybody. So in summary, food availability doesn't impact the microbead ingestion. It's just how many microbeads are there. Larvae reach ingestion saturation at very high concentrations. So this means as the microplastic pollution grows, they will continue to ingest more. Microplastic ingestion thresholds, so where they start to ingest those particles, are from about 10 to 100 particles per milliliter, and that's based on size. So if they're a larger size, it has to be higher concentrations for them to begin to ingest. We see very slight effects on physiology, so those are with the fibers at very high concentrations. And I mentioned we did nylon and polyester. All the nylon larvae died, so it was very hard, all of them, so even the controls, so it was hard to kind of get any information from that, but it would be great to do that again. So overall, microplastics show no to low impacts on oyster physiology at environmentally high concentrations. So the current maximal environmental concentrations are estimated to be 0.1 to 1 particles per milliliter, and that's sort of in coastal regions globally. The most recent survey in the Chesapeake Bay was that there was about 1.1 particles per liter, so about 1,000 times more dilute than in other places, and much lower than where we saw the beginning of ingestion for larvae. But there's a caveat, and that's we don't have very good methods to measure these particles that are small enough to be ingested by these small organisms. And so it's important to know that in the future, as methods get better and we can measure these particles better, this might change. And also, microplastic pollution is predicted to increase too. So in the near future, this could also change. And with that, thank you.
Hello, my name is Todd Toman. I'm from the University of Delaware. I'm a PhD student under the advisorship of Dr. Tobias Kukulka, and this study has been performed in conjunction with Dr. Jonathan Cohen and his PhD student, Hayden Betcher. So today I'm going to talk about the transport of marine microplastic debris in Delaware coastal waters and the implications for buoyant zooplankton. So our labs for the past several years have been working to both quantify microplastics in the Delaware Bay, as well as study the factors that organize them and really set their distributions throughout this system. This figure on the top right shows a snapshot of microplastic distributions in April 2017 at a set of, I think it was around 17 sample stations. You can see high microplastic concentrations near the Delaware River source waters, as well as near the St. Johns and Murderkill River source waters as well. But the big takeaway from this plot is how inhomogeneous the distribution is. You can see towards the New Jersey coastline up to the northwest, there's very little microplastics in this particular snapshot, whereas you see higher concentrations on the Delaware coastline. But while this picture may create an image that you have essentially high inputs at source waters and then they just disperse and distribute as they move throughout the bay, it's not really that simple of a picture. Once they're in the bay, they're subject to estuarine circulation and whatever surface flows that are happening in the system, assuming they're buoyant microplastics. In this image, you can see this is taken sort of on the upper channel near the Delaware River outflow. There is a white band. I hope it's visible. I can't really see from this angle. So there's a white band that traces down the center of the channel there. And what that is actually a collection of bubbles, microplastics, organic debris, and other surface advected particles within the system. And that's being generated by the surface flows essentially coming together and forming a convergence zone that traces down the length of the estuary. So we have not just these large-scale overview patterns from the previous figure, but these smaller, fine-scale aggregation features that are generally a couple meters in width. So with this idea that microplastics aren't just evenly dispersed throughout the system, but locally aggregated, it creates this question of how do these aggregations alter the impact on biological organisms such as zooplankton in the system. And that's going to depend on whether or not the two are actually in contact with one another. So for this study, we use as a sample organism Atlantic blue crabs, or Calonectus sapidus, and we focus on their larval migration, so that's those bottom two there, the zoa and megalope. And these organisms will migrate within the water column. Early in the life cycle, they're largely surface trapped. They use their limited mobility to stay within roughly the upper 1 to 2 meters of the water column. And they actually have a very interesting life cycle in that they're born in the bay, but they actually migrate along the coastal shelf during their lifespan and passively return to the bay as a result of surface currents. So they're born in the mouth of the bay, and they're exported through river discharge that traces the Delaware coastline and then moves to the south once it exits the bay. And then winds push them back to the north up towards New Jersey. And then when those winds reverse, it actually pushes them back towards the Delaware Bay and up inside where they can again settle and recruit to the fishery. So in order to understand whether or not the microplastics in the crop are a major factor in the health of blue crabs. We have a goal of completing a risk assessment for this species based on 2 factors: the exposure levels and any adverse effects, such as for the oysters in the previous talk. So this talk is going to focus on the first half of that, determining the exposure, which can be done either through field sampling to determine whether microplastics and larvae are actually in the same place, or our approach is more model-oriented, creating a regional model for the Delaware Bay and actually advecting tracers to represent both organisms. So our research questions here are: how does floating microplastic debris organize in the Delaware Bay? Where are they and how do they get there? As well as, do zooplankton actually come in contact with these aggregations of microplastics that we can observe in nature? For this study, we use the ROMS model, the Regional Ocean Modeling System. We model the Delaware Bay and the coastal geography, and we use realistic tides, observed winds, and observed freshwater forcing to create realistic flow fields. And then we can put in surface trapped Lagrangian tracers to represent our microplastics and our larvae. And we do this based off field observations. So our observations from that April 2017 figure showing the source regions being the Delaware River and near the St. John Merrill River. And then the larvae are placed near the mouth where they would traditionally spawn and are deflected towards the Delaware coast so we can really catch that outflow of river water. So hopefully okay. So I'm going to show just a sample figure of what we get out of our model. The background here is the salinity field, and then the magenta dots here are our microplastics. And this is going to show a 2-week time slice in which we have a wind reversal from downwelling favorable, so to the southwest that forces waters onshore, to upwelling favorable to the northeast that forces water offshore. So you can see as this starts that the particles are largely aggregated towards the Delaware coastline, and that's similar to what we saw in the field observations. But over time, as those winds sort of relax to a more neutral state, you'll see the particles start to shift back across the bay. Should be about, here. So they drift back across the bay into the central channel, and then eventually it reverses to the upwelling winds, and you see they move towards the New Jersey coastline. So the big takeaway is the major driver setting the dispersion of our particles in our model appears to be the actual surface winds driving their transport across bay. So I'm going to just zoom in on a couple snapshots here. So first off, the low wind case is quite interesting in that you would expect without winds, they would generally follow the river outflow that inputs these particles into the bay in our model. But instead of tracing down the Delaware coastline, they actually aggregate in the center of the bay. And in doing so, they overlie the deep central channel in the bay's interior. And this is actually a result of density influences as the tides are running into the system. So as the tides flow in on the flood tide, the shallower flanks actually slow down the ingressing tide. So you get this dense bulge into the center of the bay, creating a lateral density gradient. And that dense, heavy core at the center of the channel actually sinks down and is filled in by waters converging from the sides. And that's actually carrying our particles out of the buoyant plume, the buoyant river plume, and into the center of the channel, as we're seeing here. And as a result of that, we're actually getting those aggregations, those tide lines I showed you at the beginning of the talk. With winds, it's a bit simpler of a picture here. When you have winds oriented perpendicular to these tide line structures, the particles tend to follow. When you have downwelling winds, they get pushed towards the Delaware coastline into the river plume, where they're rapidly flushed by the outgoing river water. And then when you have upwelling winds, they're pushed out of that central channel and up towards New Jersey, where you don't have as much outflow, so particles can hang out a bit longer. And they tend to spread out a bit more because this has a bit more of a complex bathymetry with several shallower channels along the bottom, creating a similar sort of effect to the main channel. So with this image of winds basically setting the microplastic locations, we can now ask what happens when we add larvae to the system, and will we get any sort of overlap between the two? And we do end up seeing that. So I'm going to focus on two of our largest exposure mechanisms within the bay. So on the left here, you have, again, the same picture of downwelling winds driving microplastics to the Delaware coastline, and then the black particles here are our simulated larvae. You can see that they've been flushed out by the river plume and congregate along the Delaware coast on the shelf. And then on the right here, the background in this case is the larval concentration, whereas our contours going from white to black and then to red indicate increasing larval or microplastic concentrations. And you can see where we have these increases in larval concentrations, we also have increases in microplastic concentrations. So the two are being directly connected in this system. And what's happening here is essentially the downwelling winds move the particles into the river plume, and that river plume drags them over our spawning grounds. So the larvae are actually being spawned into more polluted waters, and these then travel together throughout their life cycle, increasing the exposure over time. Upwelling winds, by contrast, are more time dependent. As I showed you before, if you have upwelling winds for an extended time, the particles move, or the microplastics move towards the New Jersey coast. But if you have upwelling winds onset with larval release, that motion in the microplastics is weakened or offset slightly by the convergent effects. So it can be slowed down enough that your larvae can actually be moved north at a quicker rate and enter into that central channel and tide line, creating a secondary exposure event. And these exposure events do end up being rather important in terms of cumulative exposure of larvae to microplastics in these systems. This red line is a base level taken from field sampling studies in 2020. Basically, we took our lowest observed microplastic level and set that as our baseline for when microplastics aren't being strongly aggregated. And then for this black line here, that's taken from our model to show what happens when you have those aggregation regions. This is a sample crab larvae in our system. So you can see there are two regions of very high exposure in this time series. At the beginning, as a result of downwelling winds moving microplastics into spawning grounds, and then again, as the larvae circles back into the bay, it actually can be stored within that central channel until such time that develops and is able to settle and recruit to the fishery. And that's going to create a secondary exposure in this time series. On average, this results in a cumulative net exposure over their life cycle of about a 400% increase relative to this background level. So it can be pretty significant. So in conclusion, we have two primary mechanisms for generating exposure within the bay, either transport of zooplankton to the main channel or collection of the microplastics in the river plume waters. And these aggregations are pretty significant over time and could theoretically be important in determining the harm to this crab species. Our risk assessment so far is focused on the exposure, and we still need to group this together with adverse results studies being handled by our colleagues. Thank you.
So I may have missed this. This is for Christine. How large are the larvae? And you talked a lot about ingestion, but you didn't say anything about excretion.
Yes, so the larvae are, a fertilized egg is about 40 microns.
Thank you.
So a fertilized egg is about 40 microns, and from there they go up. By the time they're done the larval period, they're about 300 plus microns. So that's in 2 to 3 weeks they'll grow that. I didn't talk about depuration. That was your second, yeah. And so I looked at that too over a 24-hour period, and they do get rid of, and that's just with the microbeads. That hasn't been done yet with the fibers, and that could be different. But with microbeads, they get rid of a lot of them in 24 hours, but not all. And I think that's a bit size dependent too. But Adult oysters do get rid of a lot of their particles pretty quickly, and I know in Europe they require a 24-hour for adult oysters for sale. They require a 24-hour sort of depuration period for all types of any types of particles that they ingest. So I'm not sure if we require that here or not. If someone else can answer that, but.
Thank you. Any other questions?
Well. I apologize if I missed this when you said it. Hang on a second. Wait a second, I wrote it down, I forgot as soon as I got the mic. You talked about how the higher concentrations in the water, they ingested more. Did it seem like it was accidental ingestions, or were they just feverishly eating everything that was around them?
It's hard to tell because as larvae, they just want to eat and grow. You know, that's their thing, and that's what they're focused on. So I didn't look, hard enough. There's different ways that you could kind of watch an oyster larva over time as it feeds, and that would be really interesting to see that. But it seems like they're more indiscriminately feeding. The other thing could be that since they were exposed for 24 hours, bacteria and other things could have grown on the surface of that, and they could see that, as food too, and they might be able to, especially with the way that they eat, with the grinding and the style, they might be able to get something off of the particles too. I don't know.
And another question. I saw one up here.
Great talks. I have so many questions. I'll restrain myself. Did you say that? Did I hear correctly that we think that there's almost like an order of magnitude less concentration of plastic particles in Chesapeake Bay than what we know from other parts of the world? And I'm guessing we may not be able to characterize that in terms of comparing to Delaware Bay, but do you trust that data? It says that?
I trust some of the data. It's hard. It's definitely because there's such a variety of methods and standards for data, and even in published papers, you really have to dig to see and try and decide what what you trust, because there's a lot of data that I don't. But I think that from what we see globally, Chesapeake Bay seems to have less pollution, and I think the biggest sources of microplastics are larger plastics, and we seem to have less than San Francisco, than other parts of Asia, than other places around the world. So from what has been measured, and I do trust the people who have done it in Chesapeake Bay, it is a few orders of magnitude less than other places. Again, most of that is larger than 330 microns, so the 3 studies. There's only one that goes down to, I think, 50 or 10 microns, but even that, our methods just aren't very good for that. So it's really hard to understand the smaller microplastics and the concentration. So I think it will change as our methods get better. I think we will see more.
Sure, real quick.
And just to clarify, my question wasn't like, was it bad science? More, is it overstepping the bounds of like what we can infer from one place to another? But do you think that from what you found that it's maybe part of the natural history of oysters and their evolutionary history to ingest a lot of inert particles, and maybe that the plastics, as long as it's inert, it's probably the numbers that we're talking about, it's similar. The effect of the plastic is similar to a little speck of sand or something in their diet? Yeah,
definitely possible. Oysters are used to handling a lot of different types of particles, sand, silt, anything. And so I think that they're in particular good at getting rid of those particles and handling them versus maybe some other types of organisms. But residence time in the gut is important. I'm the only one has looked at anything besides microbeads and oyster larvae, so with microfibers, so we have no idea really how they would get rid of these differently shaped particles. In the larvae, there's been studies on the adults, and they have more trouble getting rid of sort of long fibers.
I'm guessing starvation could be an issue.
Yeah, it could be. So, thank you.
We're going to go ahead and get started with our next session on human implications. And during this session, we're going to have 2 recorded presentations, one from Carol Adrian Smith of Morgan State University, and then Britta Beckler, the Associate Director of Ocean Plastics Research at the Ocean Conservancy. We'll go ahead and get started.
Hello, I'm Carol Smith from Morgan State University, a PhD graduate student, and I'm looking forward to sharing my research with you. The high content of degraded microplastic volatile organic compounds in the Cursora chesapeake of the Chesapeake Bay, Patuxent River, Maryland, and its relationship to the aquatic and human food web. Some background. Microplastics in the Chesapeake Bay. Plastic debris degrades into smaller pieces, but microplastics are less than 5 millimeters in length and found in every habitat on Earth. Researchers have found high concentrations in and near the Chesapeake Bay from another lab, with fragment, films, and fibers being the typical morphologies, with polyethylene and polypropylene being the most abundant plastic types. Here you see 2 pictures labeled A and B, and these are the microplastics collected from the Chesapeake Bay that contain a variety of constituents, being pellets, cylinders, synthetic fibers, plastic petals, thin flexible sheets, and extruded polystyrene. How small is a microplastic? What is less than 5 millimeters compared to an inch? Here you see a ruler blown out to scale, and a red line here indicating where 5 millimeters lies in relation to 1 inch. My research focuses on 5 millimeters or less of microplastics. The dangers of microplastics: they can contain a wider range of contaminants such as DDE, polychlorinated biphenyls, PCB, and some metals. They can remain for long periods, cycling between air, water, and soil. These chemicals can leach from the plastic surfaces into marine animals, increasing the potential for toxic effects such as being carcinogenic and or damaging DNA. A little bit about jellyfish. In general, jellyfish have lived for over 600 million years, surviving extinction. They live in low oxygenated areas and have little predators. They have no blood or lymph system and cannot necessarily reject foreign objects. They are also not able to move away from danger, so they must wait for more favorable conditions before continuing their life cycles. There are over 3,000 types of jellyfish, and they're being discovered every day, new types of jellyfish. The Cursora chesapeake scientific classification is listed here, the genus and the species, and the binomial name Cursora chesapeake was created by Pappenfuss in 1936. The Cursora chesapeake reproductive or life cycle, they are sexual and asexual. Fertilized eggs hatch into round ciliated larvae called planulae, which swim and attach to the any sheltered objects. Then the polyps will develop, okay, and then the polyps develop into strobila, and through strobulation, there's an asexual process where a phaedra are created, and then you'll see that the jellyfish, as we're so familiar with, become present. Life expectancy is likely between 6 months and 1 year in the wild. Here is a picture of an idea. These are moon jellies, though. And these moon jellies here are from the Long Island Aquarium lab. And you can see that these polyps that have hooked on or latched onto something look like little tiny bubbles. So they go from being very small to the adult medusas, a jellyfish, which you can recognize today. So this is a moon jelly, which goes through the similar process as the Casora chesapeake. The Casora chesapeake's gelatinous tissue is something I focus on entirely. So with the Casora chesapeake's gelatinous tissue, it is made of at least 95% water. Its gelatinous tissue enables it to have a special ability to survive and thrive in polluted eutrophic waters that have very little oxygen. Its gelatinous tissue, as I mentioned, is made up of at least 95% water, but also polysaccharide compounds, collagen peptides, and amino acids. It seems unable to voluntarily release content caught in its thick gelatin-like tissue. So, which leads me to my central hypothesis that the Casora chesapeake will contain a large abundance of microplastic hydrophobic particles due to their natural attraction to each other and the predominantly hydrophilic characteristic of the jellyfish body. Some of my specific objectives are to qualify and quantify the presence of microplastics that can be ingested by these jellyfish species, either directly or indirectly through trophic transfer and involuntary contact with seasonal jellyfish blooms. To identify the volatile organic compounds or the microplastics from within the Casora chesapeake. Taking into account their vortex movement and how these jellyfish move through the water and how do they encounter the microplastics is also a part of my research. So methods for microplastic and contaminant detections and equipment we use to study the chesapeake from the field and in vitro. We're using an optical microscope, Raman B staining, Raman spec, LED microscope, FTIR, GCMS, X-ray photoelectron, X-ray fluorescence, and LCMS. Some data that was collected. I'd like to first show you the area where we collected the jellyfish in respect to the Chesapeake Bay and the Patuxent River and how they're connected to each other. The green triangle indicates one of my labs, which is Morgan State University, Pearl, which is located on the Patuxent River. And the stars define the collection areas where jellyfish were sampled from. The Raman B dye staining for visualizing microplastics in laboratory-based study, scientific publication, was an eye-opener for me. It was created by Tong and Al, and they used Raman B dye staining for visualizing microplastics, not from within a species, but just in general and a lab. So they would take certain types of microplastics, apply Raman B staining, and what was determined was that Raman B, RHB dye, was the strongest stain in order to identify certain types of microplastics. I was so fond of that method that I wanted to apply it to my studies in jellyfish, and I did, and I was luckily able to have a scientific publication come from it called The Effective Use of the Inexpensive LED Microscope with Raman blue staining to Identify Microplastics. And all these microplastics pictures are different types of microplastics from within the Casora chesapeake jellyfish. Some of my preliminary results are from the tentacle of the Casora chesapeake with an abundance of microplastics. And here you see sort of light staining of purple dots, and some of these dots are circled here. So what this indicates is that these are microplastics found within the small tentacle of the Casora chesapeake. The tentacle being 1 inch approximately by 0.5 inches. So as you can see, the microplastics seem to be caught in the tentacle of the Casora chesapeakes. Here is a picture of the mapping of the plastics we found within the tentacle of the Casora chesapeake and the different sizes of microplastics found from within the jellyfish. So they range from 0.2 micrometers to 33.33 or so micrometers, but every single one here is not necessarily mapped, but we just grabbed ones that we could, and we mapped those. I then applied Raman scatter spectroscopy at Dr. Dritschko's lab at Johns Hopkins University. It's our physics labs. And what Dr. Dritschko helped me do was, as you see below me, there's identification of a piece of microplastics from within the Casora chesapeake on a glass slide. And with the help of a library, we identified that this piece of microplastic is actually indeed a microplastic. And it's very small. And here you see also the spectrum of this particular microplastic. Also utilized was GCMS headspace and GCMS with methanol without a headspace. This is sort of an idea of what headspace is. We put the fluid is put inside of a glass container, and then it is heated up generally to a temperature, and then everything that is released as the temperature goes up goes through a fiber, and that fiber goes to the machine that identifies what is present from that particular liquid. Some of the VOC of microplastics we found from the Casora chesapeake are here, and these specific molecules are those that are from microplastics. The volatile organic compounds are specifically from microplastics, such as octanol, benzene, acetaldehyde, hexadecanol, octadecanol, ketones and alkanes. And we found many more, but these are just a few that identify that the contents within the Casora chesapeake are indeed, a lot of them are indeed microplastics. Proportionally, I broke down the two different methods of the GCMS. And what I found here below me, here you can see GCMS mass spec identifies the microplastic volatile organic compounds in the chesapeake through the functional groups. So to the left of you, you see, and labeled picture 5A here, the headspace GCMS used versus methanol GCMS. And the headspace was able to detect a higher amount of presence of microplastic volatile organic compounds. And over here in picture 5B, you can see that there was a detection of types of microplastic VOCs, but a lesser amount. This is a picture of microplastics that carry non-plastic VOC contaminants from within the Chesapeake. Again, from Dr. Dritschko's lab at JHU, Johns Hopkins University, utilizing Raman spectroscopy. And here you see a microplastic sort of shaped like a big banana. And in the middle is a red dot, which concentrates the reading of some of the contaminants on this particular piece of microplastics. And microplastics from this particular, this microplastics that had a contaminant on them other than VOC microplastic contaminants, the contaminant within this microplastic that is not a microplastic contaminant is tetrachloroethylene. So again, these pieces of microplastics will come from within the Chesapeake. Now, we did not only find tetrachloroethylene. I'm just showing you one of the contaminants we did find on the microplastics from within the Chesapeake. The dangers of tetrachloroethylene to humans is that tetrachloroethylene may harm the nervous system, liver, kidneys, the reproductive system. It may be harmful to unborn children and poses a higher risk of getting certain types of cancer. Why is this important? And how do jellyfish contaminants become a part of the human food web? Microplastic contaminants of the jellyfish move through the human food chain because the jellyfish are consumed by fish and crabs, which humans frequently eat. Moreover, humans consume jellyfish directly. They can consume human fish directly. Some cultures have a direct human consumption of jellyfish. For example, here are some jellyfish dishes. We have a simple jellyfish salad, jellyfish with shredded chicken at a restaurant, a jellyfish roast duck, a Korean dish incorporating jellyfish, a jellyfish salad again, and a close-up view of Cantonese-style jellyfish. However, in the case of the Chrysaora chesapeakii, the predators of Chrysaora chesapeakii, there's a relationship. So Chrysaora chesapeakii contributes to the aquatic food web of the Chesapeake Bay. They function as strong contributors of microplastics into marine trophic webs, which are passed into humans via the predators, the jellyfish predators, because of being fish and crabs. So for example, here is an aquatic food web of the Chesapeake Bay and Long Island Sound created by me. And on the bottom of this food web, you can see that the microplastics and volatile organic compounds are then consumed by the jellyfish, which are then consumed by fish that are commonly eaten by humans, such as flounder, tuna, swordfish, shark, snapper, and crabs, certain types of crabs. My research exposes the Chrysaora chesapeakie's vulnerability to drifting microplastics of volatile organic compounds within the Chesapeake Bay, Patuxent water column. And the Chesapeake contributes to the aquatic food web of the Chesapeake Bay and serves to carry microplastics through to higher marine animals, which are then consumed by humans. Additional studies would contribute a greater understanding of trophic transfer, released, or ingested microplastics from the Chrysaora chesapeakii and other jellyfish, which are I'm now looking into, such as lion's mane, comb jelly, and the moon jelly. Here are my references. Thank you so much. Here is my publication. The effective use of the inexpensive LED microscope with the Raman B standing to identify microplastics, which specifically focuses on microplastics from within the Chrysaora chesapeakii. This is the end of my presentation, and thank you for attending my presentation. This work was from Morgan State University, Dr. Promanik's lab, and Pearl University and Morgan State University. This research is supported by the National Science Foundation, grants acquired by Dr. Fand, the head PI, and Dr. Dritchko of the Johns Hopkins University.
Hi everyone, Dr. Britta We're here with Ocean Conservancy. Excited to talk to you today about two studies our team has been engaged in regarding microplastics in the US. One is a social survey and the other is a lab study. So how do these two things connect? We know that microplastics are a key facet of the microplastic pollution issue, unfortunately. And we do need more investigation of the human dimensions of the issue with respect to social science and human exposure estimates, because we don't know the human health risk of exposure to microplastics yet, and we need to know what our exposure levels are first, because part of that risk calculus is our exposure as well as impact. And to reduce microplastic pollution effectively, and hopefully our exposure as well, we need to understand and characterize what the public knows, what they perceive, and what solutions they support in tackling the microplastics problem. So first up, I'd like to talk about a study we conducted with our partners at Edge Research. My co-authors are listed here, about microplastics and plastic pollution here in the US. And just a note, this and the other study I'm going to be talking about are undergoing peer review. So if you don't mind, please refrain from screenshotting or sharing this information with others until it's hopefully published very soon. So the design and approach for our social survey, it was conducted via a national research panel in fall of 2021. We wanted to get at that public perception question. We wanted to know awareness, concerns, and views on some ocean issues specifically. But for this presentation, we'll focus on plastic pollution and microplastics and support for reduction measures tackling plastics. And we also wanted to determine consumption of several different protein types that we later study in the second piece I'll be talking about today for microplastics to get at that human exposure question. So we conducted the survey and had about 2,000 respondents, and the responses were managed so that we got a US Census representative sample. So the results we have are representative of the US population writ large. The first question, it's not related to microplastics necessarily, but I do think it helps kind of color the responses later on in the survey. So the first question we asked was about perceived ocean threats. We said below is a list of problems that could impact the ocean or how people interact with it. For each, please tell us if you think it is. And we provided these options. Results there, I'm showing only those that identified as either not much of a problem on the left in gray, a pressing problem for today, or a problem for the future. And there were some interesting takeaways here. The public recognizes many issues affecting the ocean in the immediate term or future. Plastic pollution was seen as the most pressing issue, but other pollutants like oil and chemicals were also top concerns. And as you can see, climate change actually lagged. Its impacts were listed as the 6th greatest ocean threat. The next question we asked, if the respondent indicated that plastic pollution entering the ocean is one of the pressing problems facing the ocean today or will be in the future, they were asked to rank their top 2 concerns about plastic pollution in the ocean. And we provided this list of 12 impacts of plastics. Impacts to marine animals were the primary worry when it came to plastic pollution in the ocean. Other top concerns were the sheer volume of plastics and, interestingly, the chemicals they emit. And just to note that 91% of US adults recognized that ocean plastic pollution is the most pressing ocean issue. Now focusing specifically on microplastics. The first question we asked was really basic. Have you ever heard of the term microplastics? And about 50% of the US adult population had. We then asked how familiar they were with the term microplastics on the top right, and then how concerned are you about microplastics in the environment and about microplastics that humans are exposed to in day-to-day life. So as you can see, about 18% were very familiar with the term or concept of microplastics. And if you look at the bottom right panel, concern about microplastics in the environment actually edged out concern over human exposure for folks that responded that they were very concerned about microplastics. But the opposite was true for those that were only somewhat concerned. We then asked, how significant a threat do you think microplastics are to? And we provided this list of items on the x-axis. And this figure shows responses of adults that said very or somewhat significant. Sorry, the figure legend got cut off, but the dark blue indicates very significant and light blue indicates somewhat. And as you can see, concern is fairly high across all of the different environments as well as for humans. Though it is notable that less than half of the adult population thinks microplastics are a significant threat to human health or terrestrial ecosystems or terrestrial animals. At this point in the survey, we provided a definition of microplastics to the readers so we could kind of do a before and after level of concern. So after providing this passage here, we said, reading this, how concerned, if at all, are you about microplastics? So here were the results, with the before responses being in dark blue, the after responses in the lighter blue. And respondents did express concern about microplastics before they were provided with the passage, but there was an 11% increase in those reporting being very concerned after they read it. The next section of the survey was about taking action on microplastics. So we asked, below are some concerns that have been raised about plastics and microplastics. For each one, how much does it concern you? And while I know these are hard to read, the takeaway here is that we provided a range of concerns from persistence in the environment to human exposure to presence in animals and plants. And none of these concerns really emerged at the top. For the most part, they're all equally concerning to the US public. But that being said, persistence of microplastics in the environment, as in they are impossible to effectively clean up, was the top concern, while chemical pollution from plastic production ranked lowest. Here we asked, who do you think has the most responsibility to reduce the use of plastics and prevent microplastic pollution from increasing? We see here that the US public places the most responsibility for action on industry, the plastics industry, which we defined in the survey as producers and manufacturers, followed by government, then consumers. And interestingly, when we investigate responsibility for reduction of microplastics at different levels of government, significantly more responsibility is placed on the feds over state and local governments. Here we wanted to know about level of support for different policy actions. So we asked, below are some actions that could be taken to stop the amount of microplastics entering the environment, water supply, and ocean. For each do you, and we asked about their level of support. And these policy actions characterize a range of different options, from oversight on plastic pellet discharge to the Break Free from Plastic Pollution Act to washing machine filters, etc. And we're only presenting responses for strongly support and somewhat support in this figure. So we were excited to see broad support for most regulatory actions, but there was the least amount of interest in the government study or personal involvement in a local campaign. Proposals to prevent discharge of microplastics into water systems and reducing single-use plastic all garnered roughly equal support. And it was really nice to see strong support for most of these actions, surpassing the 50% mark. The last question in the survey that I'll be talking about touched on individual action. The question was, below are some actions you can take personally to reduce your everyday plastic footprint and the likelihood of plastics leaking into the environment. For each would you. Here were the options, including I already do this frequently or occasionally. And so these are the results for items people reported already doing frequently or occasionally, or also indicating they definitely would do. So up in this top tier here, recycling and using reusable bags and water bottles are the actions that people reported doing consistently. There were significantly more people who reported they do these than any of the other actions presented. Below those were some actions that may be less familiar to folks or harder to execute and may take more prompting or support. And political and consumer advocacy ranked lowest in terms of uptake. Those were the last two items on the list, kind of lagging behind the other actions. So that was the social survey, a quick take of the social survey. Now we're going to segue into this lab study where we took some of the social survey data on consumption of different proteins and actually paired them with lab results to get at the human exposure question. So thank you to University of Toronto and my colleagues on this paper as well. So the research questions we were asking with this body of work: what are the microplastic burdens in different protein products here in the US? We know that protein represents about 15% of the American diet, so it is significant, and it's underrepresented in current microplastic exposure estimates. We wanted to know, do microplastic burdens vary based on the type of protein, the level of processing, brand, or the store type? We wanted to know the influence of packaging on microplastics in these products. And lastly, the annual adult exposure to microplastics from consuming those proteins. And that's where we plugged in our social survey data. So for this study, we had 16 sample types. We had unprocessed seafoods, which were obtained directly from vessels. We had minimally processed packaged products and highly processed packaged products that were all purchased directly from grocers here in Oregon, where I live. We purchased multiple brands of each sample type. We had a conventional grocer and a natural grocer that we compared products from, and we had multiple replicates of each product. So we were able to see if there were any differences in microplastics. Here's myself and Maddie, a colleague in the lab, chemically digesting our samples, watching them break down so that we were able to isolate microplastics, sieving the samples, looking at them through a microscope, picking out suspected microplastics, and then running those through a Raman or FTIR to chemically validate the material type. We had a number of quality control measures, including those to reduce contamination into samples. We used a blank correction method, and we wanted to ensure that we were not overestimating the amount of microplastics in our samples. So what we found is microplastics, not surprisingly, in 88% of all of our samples. Looking by product, the average number of microplastics per serving was 72 across all products, was lowest in chicken breast at 2 microplastics per serving, highest in breaded shrimp at 366 microplastics per serving. And in looking at microplastic category, fibers were the most prevalent. Of all the suspected particles we encountered, 94% were actually microplastics. So we then spectroscopy, can never say that word, corrected our data to remove the other 6%, which were either identified as natural or unknown materials. We found lots of different plastic polymers, including rubber, semi-synthetics, as well as particles with synthetic additives and dyes on their surface. In terms of differences we observed, when looking at processing level, we found that highly processed samples contain more microplastics than minimally processed samples. However, we did not detect any other significant differences when we looked by protein type, so seafoods versus terrestrial meats versus plant-based proteins, or by brand, or by grocer type, or conventional grocer versus the high-end grocer. We also saw very little evidence, if any, of packaging-derived microplastics being found on the product specifically. For example, if something was packaged in clear, flexible plastic, we would only see perhaps 1 to 2 matching fragments of that polymer type on a sample. So we did not think that this was a significant correlation. In terms of human exposure, we used our social survey data, the consumption information, and the lab-based information to get at a range of exposures, which you can see here. So most interestingly, in terms of annual US adult exposure, you can see an average as well as a range. If you're eating 1 serving of this protein per month or 10. So on average, if you're consuming breaded shrimp, which contain the most microplastic of all of these proteins, you'd likely be exposed to about 13,000 microplastics per year. However, if you're consuming chicken breast on an average level, you may only be exposed to about 140 microplastics per year. So a huge range and a lot of deviation, just because that's the name of the game with microplastics research. There's a lot of variation from sample to sample. So putting these results into context, previous studies have estimated adult microplastic exposure is about 320,000 microplastics per year. Through the proteins we studied, we estimate those contribute about 11,000 microplastics per year. But if we're using the highest microplastic values across our sample types, that could be as high as about 3.8 million microplastics per year. So in conclusion, looking at these 2 studies taken together, plastic pollution is seen as a #1 threat to ocean health by adults here in the US. 50% of us have heard of microplastics and have concerns. If you provide information, concern increases. Americans think the plastic industry, manufacturers, etc., are most responsible for reducing microplastic pollution. There's broad support, it appears, for regulations to reduce microplastics in the environment. When we look at our foods, all of the US sourced proteins we studied contain microplastics. Highly processed products contain more than minimally processed products. And it goes without saying, but we're exposed to microplastics through the foods we eat. There's definitely more research needed to gain a more complete picture of our exposure to microplastics and the human health risks associated with it. That's all I have for you. Thank you very much.
So your data showed that the breaded shrimp had a significantly higher quantity of microplastics in it. Did you test the breading out of curiosity to see if that's where the microplastics were coming from in a majority, or do you really think it was from the shrimp?
And pause as we translate the question. I was wondering that too. Because lots of the other shrimp it didn't seem to be a shrimp thing. It seemed to be a breaded.
It all seemed like an issue, but the breaded was very interesting.
Yeah.
So Britta says that's a great question. And she said, we did not. We just digested the breaded shrimp whole.
So my question was actually about the public perception of it, because I noticed that the public seems to think it's industry and government's problem to fix, and yet there seems to be a high percentage of people that are doing things like using the reusable cups and the bags. So do we think that we've worked against ourselves by promoting reusable cups and bags as opposed to talking to people about community-wide solutions or lobbying industries? Like, do we think that maybe our push to get people away from the plastic bags may have hurt us in the long run based on that perception?
Okay, so from Britta, we have an answer back now. She says, oh gosh, that's a great question. She says, I don't think it has. People are willing to reuse bags and bring reusable items more than any other action we presented.
So I was just wondering if she could tell us about the geographic distribution of the survey respondents, because I know she's located on the West Coast, so I'm wondering how distributed the survey was.
Yeah, she said the survey was managed to reflect the 2020 U.S. Census, so it was a national representation.
So now we're going to transition to our last portion of the microplastics and nanoplastics session. And during this one, we're going to be talking about wildlife implications. And we have Hayden Bosher of the University of Delaware and Bob Murphy of Tetra Tech. Hayden, if you want to come up.
Okay. Well, thank you for having me. This is such a treat. I've been writing my dissertation for the last 2 months, and I'm running out of things to talk about with my cats. So it's nice to talk to other people. I'm looking forward to having some conversation after the presentation. My name is Hayden Boettcher. I'm a graduate researcher at the University of Delaware, where I spent the last 5 years researching marine debris, specifically on crab larvae. And so today I'm going to be presenting my research on the effects of polyethylene microfibers on Panopeus herbstiae larval development, so focusing specifically on the larval development side. Okay, so for those of you who haven't seen a crab larvae before, this is what they look like. They look nothing like adult crabs, but they're great nonetheless. I've spent a lot of time looking at these guys under a microscope. And so we're trying to answer this big question, right? What are the effects of microplastic exposure on crab larvae? And so we tried to answer this question using two different types of experiments. The first one was long-term developmental experiments where we exposed crab larvae over the entirety of their larval development. That takes about a month. And the treatments that we used were polyethylene terephthalate microfibers. We actually have seen evidence of natural ingestion of these microfibers, which my friend Miranda is going to be presenting in one of the posters over there later on. So be sure to check that out. And so we exposed these crab larvae to PET microfibers at 4 different concentrations. The first one was a control. It was 0 microplastics per milliliter. And then it kind of ramped up from there to 0.1, 1, and 10 microplastics per milliliter. And during these month-long experiments, we measured larval survival, their development time, and their body size. And then as a follow-up to these developmental experiments, we also did grazing and respiration experiments. And here we switched up the treatments a little bit in that we used microplastics, but also natural sand microparticles, which is something I think I've heard people in the audience question a little bit, which is, are the effects that we're seeing due to plastics specifically, or just particulates in the water, right? Because organisms are experiencing natural particulates in their environment as they grow anyways. And so for these experiments, we had both microplastics and sand of the same size at 10 microplastics or sand particles per milliliter, which is high, but if there were any differences in effect, we wanted to be able to see that. So that was kind of a concession we were willing to make. And the endpoints we measured here were grazing and respiration rates. So first, I'd like to talk you through the developmental experiment setup. I'm going to focus your attention first to this bottom kind of timeline here. Crab larvae don't grow like you and I grow. They grow in very distinct stages. And so they work their way through the Z1 stage all the way through adult crab. And we're focusing specifically on the Z1 through megalope part of this timeline. And we ran 3 different experiments because we wanted to toy with the exposure duration as well as the concentrations. And so in experiment 1, we had a full exposure experiment. We kept microplastics in the treatments for the entirety of their development. Experiment 2, we only included microplastic exposure in the first 3 stages and then took it away after that to see if recovery was maybe a possibility if there were any effects. And then in experiment 3, we did the reverse of that. We kept them in their control conditions for the first 3 stages and then included microplastic exposure after that as a way to see if maybe early stage zoea might be more affected than late stage or vice versa. And I wanted to kind of give you an idea or a visualization of what the experimental setup looked like as well, briefly. And so we used these 1-liter glass jars, which housed 30 individual zoea each, as well as food and microplastic concentrations. They were kept on this rotating plankton wheel to keep everything in suspension, and the glass jars were sealed. So oxygen availability could become an issue, but we did water changes every other day to prevent that. And so water changes and data collection were done every other day for an entire month for each of these experiments. This was able to keep the water fresh, keep the microplastic concentrations consistent, and the amount of food consistent as well. In total, that came to about 90 individuals tested per treatment. And so first I'm going to talk about the impacts on survival. The big takeaway is that any impacts on survival are relatively inconsistent, if there are any. I'll walk you through this plot starting from left to right. We have survival probability on the y-axis. This is a measure of Here we go, I got the cursor here. So at the start of the experiment, you're at 100%, right? Everybody's still alive. This tends to taper off over the 25-day exposure period. And in the full exposure experiment, though it looks like there may be a little bit of separation here between the low and control microplastic treatments, this gray and pink color, and the high microplastic treatments, the darker pink and the red. The variability that we saw in the replicates, which I'm not showing here so that we can actually see the lines, was overlapping quite a lot. And so there were no significant differences there. However, in the early exposure experiment, we did see a significant difference in survival between the control treatment and the low microplastic treatment, the 0.1, and the two high microplastic treatments, the 1 and 10 microplastics per milliliter. So we thought, okay, maybe we're onto something there, but we saw the opposite in the last experiment. And so in the late exposure experiment, we saw that the 0.1 treatment survived less than the other three. And so what my takeaway from this is larval survival is widely variable in a lot of species, particularly in crab larvae. Any effects that we would be able to see, I think, are just being washed out by overall variability. But that's not to be, the same isn't to be said for body length, which we measured. And so here we saw that high exposure levels actually caused a decrease in larval body length. Again, I'll walk you through this plot. We have body length in millimeters on the y-axis, basically getting bigger as you move up the y-axis. And so what we observed was that zoea in the control treatments are growing bigger, particularly in comparison to the two highest microplastic concentrations, the 1 and the 10 microplastics per milliliter. And this was consistent when measured at each of the subsequent developmental stages. In the early exposure experiment, we saw the same thing, but we did actually see the possibility of recovery once microplastics were removed from the system. And we saw kind of the opposite thing in the late exposure experiment, where there were no impacts until microplastics were introduced, and then we saw the same consistent drop in body length. And these decreases in body length aren't huge. Just to note the scale of the y-axis, it's only a 3 to 5% decrease in overall body length, but it's still significant to note. Moving on to the developmental delays, which we observed with increasing exposure. So likewise with the high microplastic concentrations, we saw delays in development. And so again, I'll focus your attention on the y-axis here. We have median development time. This is a measure of how long the zoea are spending in each developmental stage. So it's a good way to measure how long they're taking to get through each stage. So for example, the Z2 zoea here in the full exposure experiment, the control zoea were taking about 6 days to pass that second zoeal stage, whereas those exposed to high levels of microplastic exposure were taking about 8 days. So it's about a 2-day developmental delay. This was consistent for most of the zoeal stages, and again, we saw evidence of recovery when microplastics were removed, particularly here in the Z4 stage of the early exposure experiment when the microplastics were taken away. Oops. When microplastics were taken away, they were able to recover back to a normal developmental time. So next, I will focus on the grazing and respiration experiments. The experimental setup for these was a little different in that we tested individual zoea instead of groups of zoea. And again, just to remind you, we had 3 treatments: a control treatment with no particles whatsoever, and then a microplastic treatment and a natural particle treatment, which was sand particles. And in each of these vials, we also had 10 food items. We were using Artemia nauplii, but we wanted to have a specific amount of food in there so that after the experiment is done, we can count how many are left and figure out what the grazing rate is, how many each individual zoea is eating per day. And then we were also measuring the respiration rate using these fancy little oxygen sensors here, which allow you to take oxygen readings without having to open up the vial, which is very handy. So what we observed is a reduction in grazing rate, both in the natural microparticle and the sand treatments. On the y-axis, we have the amount of food, the number of prey items eaten per day. In the control treatment, I apologize that the numbers got a little mixed up, but that's 4.7 on average eaten per day. Whereas in the sand and microplastic treatments, it dropped down to 3.7 and 2.7 prey items eaten per day, which is about a 21% or 43% decrease in the amount of food you're taking in every day. And so it's important to note here, I think, that natural microparticles do have an impact, though there isn't a significant difference between the control and the sand data. But there's a larger impact when exposed to microplastics, which is a significant difference from the control group. And speaking of respiration rates, we saw a similar pattern, except with an elevation of respiration rate. Respiration rate is a good measure of how much energy you're expending. And what we observed is that Zoea in the control treatment are around 0.26 micrograms of oxygen per individual per hour, right? This is just a measure of how much breathing you're doing. And so in the sand and the microplastic treatments, we're seeing that they're actually respiring almost double in the sand and over double in the microplastic treatments. And so again, here we see that both sand and natural particles do have an impact, but microplastics are elevated just a little bit. And so the key takeaways I'd like to leave you with are that in the developmental experiments, we saw a decrease in body size. Survival was relatively inconsistent, but it also took larvae longer to reach the next stage, in addition to the size being a little reduced. But recovery was observed once the microplastics were removed from the system. And then with the grazing and the respiration experiments, we saw that both natural microparticles and microplastics caused similar effects, though they were more extreme in the microplastic treatments. And so as you can imagine, if you're a Zoea, you're ingesting less food and spending more energy, and so you're kind of getting hit on both sides of this energy budget, which I think helps explain some of the observations we saw in the developmental experiments. These delays that we observed, I think, could have an impact on the amount of time that Zoea are spending in the plankton. The plankton is a dangerous place to be if you're a crab Zoea. Everything is looking to eat you, so the sooner you can get out of there, the better. I think the developmental delays we see could impact your mortality if you're a Zoea in the plankton, though I do think it's important to note that the concentrations of plastic we observe these effects in are much higher than what we currently see in the Delaware Bay, the Chesapeake Bay, anywhere along the East Coast. Though I think it's still useful to highlight so that we have a reason to prevent this in the future, because things are only going to continue to get worse if we don't. Lastly, I'd like to thank NOAA Marine Debris and the Delaware Environmental Institute for funding the research, and two undergraduates, Heather and Naz, for helping me complete the massive amount of work it took to do the research. And thank you so much for having me.
Our next speaker is Bob Murphy from Tetra Tech.
Thanks for having me. It's the last talk before lunch, so I think everyone will be very anxious to get out of here. So a lot of the talks so far have already set the stage for what microplastics are by definition, so I'm not going to go into that. We've had some good talks about organismal effects. So my talk is going to be on developing an ecological risk assessment for microplastics. So this is more of a conceptual, this is not a quantitative risk assessment. And we did this in the Potomac River estuary. I'd like to acknowledge my co-authors here, Jennifer Flippin with me at Tetra Tech, Ryan Woodland at the University of Maryland, and Kelly Summers, who's actually here today from EPA Region 3, who funded this work. There we go. All right, so a little backstory here. So microplastics, as we know, are an emerging issue. Didn't know much about them in the Chesapeake region, or along the East Coast for that matter. a group of us submitted a proposal to the Chesapeake Bay Program Scientific Technical Advisory Committee to put together a workshop on the issue. That's what started it all. It started a big ball rolling. So this was in 2019 we put on the workshop, and it was couched under emerging issues of concern. So by 2019, there had already been plenty in the news about microplastics, so it had emerged, but we didn't know much about it. Putting together the workshop, we actually designed it in a format similar to how you would conduct an ecological risk assessment. So it's a problem formulation. This is an abbreviated risk assessment framework on the right there from the EPA. We did a risk analysis. We had some talks about the level of risk that microplastics would have on different organisms, and then to characterize it. So very similar things like LC50 estimates, mortality estimates and things like that. Out of the workshop came a series of recommendations to the Chesapeake Bay Program. Now this is very wordy. The first one was that the Bay Program create a new team. So those familiar with the Chesapeake Bay Program, it's a fairly bureaucratic, large group. It moves slowly by definition. I've never seen it move this fast. We gave a series of talks. My co-convener, Matt Robinson, who's now with the EPA, across the Bay Program work groups and teams, and we left with a management team with a mandate to create a new plastic pollution action team, or as we call it, the PPAT. All the work that has been done in the Chesapeake region through the Bay Program regarding microplastics is being done with the PPAT's advisement. The second thing that the team recommended was to develop a risk assessment model for microplastics in the region. I'm not going to concentrate on the other three items on this because we want to really focus today on number 2 here, which is development of a risk assessment model. When the PPAT first convened, we talked a lot about how this risk assessment would look. We decided rather than focusing on the Chesapeake Bay, which is a very large body of water, large watershed with a lot of variables, we tried to tackle a smaller estuary. In this case, we used the Potomac River. It's the mini Chesapeake, if you will. And we decided on striped bass as our species of interest. There's a lot of debate regarding the decision of a species. We want something iconic. Blue crabs fit that very nicely. Oysters are great because they're sentinel. They don't move anywhere, so they could be an indicator of the body of water and the exposure rates. But we decided on striped bass and focusing on the young of year only because the Potomac River is the second largest spawning ground for striped bass on the East Coast, the upper Chesapeake Bay being the largest. And we focus on young of year because they are resident, they are non-migratory at that age. So in order to pull the risk assessment model together, we did an extensive literature review on diets for young of year striped bass. We focused mainly on the Potomac River. Believe it or not, there is some good literature on striped bass young of year diets in the Potomac. We also took studies under advisement from other places in the Chesapeake region, and as well as along the Atlantic coast. But the Chesapeake is one of the most well-studied populations of striped bass on the East Coast, so there's a good deal of literature we could rely on for that. We did look at some global estimates for numbers. I'll touch on that a little bit later, just to look at different pathways and vectors of microplastics. So there's young of year striped bass on the right there. They actually vary in size quite a bit over the course of the summer. And that's our biological endpoint of interest here. In a risk assessment, we want to look at certain endpoints. So this could be growth rates, this could be reproductive capability, predator susceptibility. We heard a little bit about that earlier. And this diagram on the left is sort of this prey network you see. As we developed the model, we wanted to do the qualitative food web interactions. There's not a lot of data available to at this stage for microplastic ingestion on a number of the organisms that striped bass are exposed to. So this is more of a qualitative effort. For example, just combining all the literature, all the studies that we decided to use, striped bass eat a lot of different things, depending on where they are in their life cycle. So we focus on the age 0, the young of year striped bass there, and you can see that just a few lines go to it. They're not eating adult menhaden, for example, at that stage. Then the semi-quantitative version of the risk assessment was looking at the proportion of prey items. So this is getting at how are striped bass going to be exposed to microplastics potentially, based on what they're eating. So the Potomac River is a very well stratified estuary. So it goes from tidal fresh up in Washington, D.C. region down to mesohaline by its mouth in the Chesapeake Bay at Point Lookout, Maryland. And because of the different salinities that the river has, striped bass are exposed to different prey items. So this was broken down by salinity regime. A lot of this data comes from Walt Boynton's work in the early 80s. I don't know if you know Walter. He used to be a fish guy before he became a nutrient guy. And they did some very nice diet work, some old school ecology, as I call it, fish ecology, dissecting out stomachs and evaluating what was inside. So in the tidal freshwater portion of the Potomac, insects dominated the diet for striped bass. These are replaced in the oligohaline. You start to move still with insects being a very large component of the striped bass diet, but then you start to see some more of the brackish water species. So mysid shrimp, for example, show up, and the thickness of the lines connecting represent the relative abundance in their stomachs. Mysids continue to be a large portion as you move into the mesohaline. Insects drop. That's not surprising. Insects are usually found in great abundance in saltier water. This is a very busy table by design, supposed to be. This was the literature review when we looked at global pathways for microplastics into organisms. Now, I'm going to admit that this is already dated. This work was completed about 2 years ago, but as we heard earlier, the number of microplastic studies is, 1,600 last year, or this year alone, so this is already kind of outdated. But the take-home is we looked at a lot of those different taxa that have been shown as prey items for young of year striped bass, and we looked at studies, whether laboratory or environmental, field studies, and all of them had microplastics. All the taxa that striped bass would feed on showed evidence of microplastic contamination. So that's both by the habitat type, so this is like SAV and epiphytes and benthic organic matter, all the way to the organismal level. So we looked at the priority prey items. Now, this is sort of the next step. What are striped bass? We saw that they're fed, or they're feeding on a number of different taxa. We wanted to look at some of the primary ones. And some recent work, it's not published, it's out for review right now by the Smithsonian Environmental Research Center in Edgewater, Maryland, did an analysis, a genetic analysis using eDNA for stomach contents for juvenile striped bass around the Chesapeake Bay. And they confirmed the 1980s work with the three primary taxa being insects, mysid, shrimp, and amphipods that comprise the large portion of the striped bass diet. So that the risk assessment conceptual model is now done has allowed us to sort of visualize and look at where striped bass might be exposed to microplastics. We pulled some numbers from the literature for these three taxa. Bay anchovy is the most common fish actually found in the Chesapeake Bay, so it's a very popular prey item for striped bass, and they range 1 to 6 particles per. Mysid shrimp, very common organism found throughout the Chesapeake and in Delaware Bay and on the coast. They range from anywhere from 1 to 38 particles per organism. Now, mice and shrimp are pretty small, so that's a pretty high dosage level, incidentally, by body weight. And same goes for amphipods, very high. And these ranges are pretty extensive here. Now, again, all of these studies where we had these semi-quantifiable numbers did not come from the region at all. So some of this work was done in Japan and elsewhere. But the point being, conceptually, there's likely exposure by striped bass. So it's that big question mark there, is what's now getting to striped bass. So that's where the assessment model has taken us. And it's actually informed some of our next steps now, where we're doing a series of field and laboratory studies. So we want to look at the loadings of microplastics naturally. So we'll be sampling for mice and shrimp in the Potomac River and the Patuxent River of the Chesapeake, and look at what the background noise is. What rates of microplastic loadings do they have? We were going to then expose, this is to getting at the quantifiable endpoint now for a risk assessment. We want to expose dosed mice and shrimp to young of year striped bass and measure any response. So the response could be liver damage, it could be slowed growth rates, it could be slowing down swimming, so more susceptible to predation themselves. So we want to look at this trophic transfer to striped bass, and we are in the process of doing that right now, so stay tuned. Maybe I'll give a talk in the next meeting to update this. But what it turned out was that the risk assessment model was very useful in focusing the discussion on these pathways. There's a strong interest in striped bass in particular, not just because of the recreational fishery, but because of a steep decline. We've seen about 6 to 7 straight years now of really poor recruitment to the fishery. Not suggesting that that's microplastics is the reason. We never know. It's loss of habitat mostly. But any additional stress on the population is certainly of a concern to fisheries managers. Excuse me. So that's where we're going to get. We're only going to be looking at mice and shrimp in this case, but we will look. We are going to make a recommendation to look at the other 2 primary prey taxa for these to get at these numbers. Okay. This was. When we looked at the risk assessment model, we wanted to put it in context of trying to capture everything that could be a source. And people who've done this sort of work before, it's really hard to capture everything in one conceptual model. Having to put a picture of the tractor in there was very difficult. But it does give the idea of, the plastics come into the environment and they end up somewhere. We learned a lot of them end up in breaded shrimp, apparently. But we're also we're finding them in fish as well. And I think I'm going to leave it at that.
And question for Hayden. Just 2 questions. How do you get Z1 size crab larvae? I'm sure you don't go to Petco. And second, how do you change the water out in your samplers without, again, sucking out the crab larvae?
You've opened up Pandora's box with that question. I really could talk for forever about that. The crab larvae we raised from egg, so we went out and collected gravid females and brought them back and waited for them to hatch the eggs and then collected the Z1 larvae. So that's how we got all of our larvae. In terms of the water changes, it was a bit of a painstaking process. It essentially involved pouring the entire volume of water in each bottle into a large glass dish and then pipetting out the zoea by hand under a microscope into a new bowl and then putting them all back. So that was my daily routine for summers on end. And so it was a little bit of a labor of love to do that, but it's the best way, I think, to do those water changes. And I think the water changes helped keep everything pretty consistent in the experiments. But I can talk for forever about the methodology of that.
I have one question, and it's kind of for both of you. You know, we in the beginning, Assistant Secretary Fiddler was talking about all the different scales we have to consider here, and zooming in and zooming out. With microplastics, inherently, you're zooming in on this. Zooming out for us a little bit, do you see where some of the work that you're doing in both of your research pursuits, how that can play into specific management actions? You know, Hayden, I know you were talking about like what happens when you take them away and how they recover, but I'm curious if that's kind of something that's in your foresight and what you might think it may be able to impact.
You mean management of plastic pollution or management of the resource?
Both and all.
I'll take that first. One of the reasons we were looking at striped bass is because of the importance of the fishery, both recreationally and commercially. And with low recruitment these days for striped bass, they're looking at everything that might affect mortality, they being fisheries managers, the Atlantic States Marine Fisheries Council for one. It's usually a dissolved oxygen issue, but if you can demonstrate that there's mortality from another, toxin, I'm not going to call microplastics necessarily a toxin, but if it's causing mortality, that allows managers to adjust the amount of catch. So there are management options that can be taken if you can get a better hand on what's contributing to mortality.
Gotcha. Thank you.
Yeah, I tend to agree. I think the research that I do is so specific that oftentimes I feel like I've got my head buried a bit too deep into the research. And so that's why I think events like this are useful. It forces me in particular to take a step back and consider the management, decisions that could be based on the research. I think prevention is a large aspect of that, as well as I think right now there's a lot of funding and research going into pulling plastic out. But I'd like to see a bit more of a focus on kind of turning off the tap before you start to work on the cleanup, or at least prioritize that. And so I'm hoping that this helps kind of push that ball forward a little bit. But I think that's what I got to say about it.
Yeah.
Thank you both. Bob, this is for you. Obviously, you've done a lot of work on this ecological risk assessment for striped bass. And it's taken, a few years because it takes a long time. How can we accelerate this process for other species and environments? Because we need to kind of learn the risks much faster than I think we can at the moment. So how do you think we can do that?
More funding. That's a great question. I don't know if I have a good answer for it, but I think, Christine, you might have been part of those original discussions on choosing a species, and for efficiency, we also chose striped bass. There's a lot of discussion went into why we chose that species. We could have chosen several, but if you understand a lot of the dynamics of exposure for striped bass, you can actually infer the exposure to a lot of other organisms because striped bass will eat those. So, for example, like crabs would be a good example that we can, although we focus on young of year, yeah, you really put me on the spot with that question. I don't know how we would accelerate it other than people tackling different ones, different organisms.
With all that we're talking about and looking into living organisms, I think more weight is being pushed now towards risk assessment and assessment models. So have you come across any model that you look at it as if it's more promising that other people can use?
So some work has been done in San Francisco Bay on risk assessment models for microplastics that are pretty advanced right now. They're probably some of the best. Different species, of course, their endpoints. Again, you can use multiple species and make some assumptions if it's not your species in particular that you might be interested in for consumption reasons. If it's another species that has a similar life history, you can certainly make some inferences. So there are some models out there that are better than others, for sure. Frankly, I'm not aware of any on the East Coast that are really fully developed yet, because we're just getting to learn a lot of the toxicity levels that microplastics may or may not present. So it's kind of hard to, it's not like cadmium or some heavy metal where we can do lab studies and go, it's going to kill 50 of these at this level. We're not there yet. We're not even close.